Myoviridae, Viridae. Viridae. Myoviridae is a family of bacteriophages in the order Caudovirales. Bacteria and archaea serve as natural hosts. There are 625 species in this family, assigned to 8 subfamilies and 217 genera. Subdivisions. The subfamily Tevenvirinae synonym, Tequetravirinae, is named after its type species Enterobacteriophage T4. Members of this subfamily are morphologically indistinguishable and have moderately elongated heads of about 110 nanometers nm in length. The genera within this subfamily are divided on the basis of head morphology, with the genus Tequetravirus provisional name T4 virus having a head length of 137 nm, and those in the genus Schizophore being 111 nm in length. Within the genera on the basis of protein homology, the species have been divided into a number of groups. The subfamily Myoviridae have virions with heads of 60 nm in diameter and tails of 135 x 18 nm. These phages are easily identified because contracted sheaths tend to slide off the tail core. The P phage is the type species. The subfamily Spounovirinae are all virulent, broad host range phages that infect members of the firmicutes. They possess isometric heads of 87-94 nm in diameter and conspicuous capsomers, striated 140-219 nm long tails, and a double base plate. At the tail tip are globular structures now known to be the base plate spikes and short kinked tail fibers with six-fold symmetry. Members of this group usually possess large 127-142 kb non-permuted genomes with 3.120 kb terminal redundancies. The name for this subfamily is derived from SBO plus Unilatin for one. The haloviruses H-phone and HF2 belong to the same genus, but since they infect archaea rather than bacteria, are likely to be placed in a separate genus once their classification has been settled. A dwarf group has been proposed on morphological and genomic grounds. This group includes the phages Aeromonas salmonicida phage 56, Vibrio cholerae phages 138, and CP-tone, Bdellavibrio phage pH 1422, and Pectibacterium carotivorum phage ZF40. Their shared characteristics include an identical virion morphology, characterized by usually short contractile tails, and all have genome sizes of approximately 45 kilobases. The gene order in the structural unit of the genome is in the order terminase portal head tail base plate tail fibers. Virology. Viruses in Myoviridae are non-enveloped, with head-tail with a neck geometries. Genomes are linear, double-stranded DNA around 33-244 in length. The genome codes for 40 to 415 proteins. It has terminally redundant sequences. The GC content is ~35%. The genome encodes 200-300 proteins that are transcribed in operands. 5-hydroxymethyl may be present in the genome instead of thymidine. The tubular tail has helical symmetry and is 16-20 nm in diameter. It consists of a central tube, a contractile sheath, a collar, a base plate, 6 tail pins, and 6 long fibers. It is similar to Tectoviridae but differs in the fact that a myovirus tail is permanent. Contractions of the tail require ATP. On contraction of the sheath subunits slide over each other and the tail shortens to 10-15 nm in length. Life cycle. Cycle. On attaching to a host cell, the virus uses its contractile sheath like a syringe, piercing the cell wall with its central tube and injecting the genetic material into the host. The injected DNA takes over the host cell's mechanisms for transcription and translation and begins to manufacture new viruses. Replication follows the replicative transposition model. DNA templated transcription is the method of transcription. Translation takes place by -1 ribosomal frame shifting. The virus exits the host cell by lyses and Holland/endolysin/spannin proteins. Bacteria and archaea serve as the natural host. Transmission route is passive diffusion. Although myoviruses are in general lytic, lacking the genes required to become lysogenic, a number of temperate species are known. Applications: because most Myoviridae are lytic rather than temperate phages, some researchers have investigated their use as a therapy for bacterial diseases in humans and other animals. Taxonomy: the following 8 subfamilies are recognized: Virinae, 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 Gorgonvirinae, Virinae, Aunavirinae, Peduavirinae, Tevinvirinae, Virinae, 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 Toragvirinae, Virinae, Virinae, Vequintavirinae. Additionally, the following genera are unassigned to a subfamily: